So my name's Erik and I spent last summer working on antisense design, which is all about regulating gene expression. So if you can create an antisense to bind to an mRNA and block the protein from being expressed, you can basically do anything. You can solve, so many problems in the world. You can prevent disease. You can do all sorts of things like that. So without this past summer, I would probably never have finished it because the academic year is so busy. But so what I found was I wrote a program that looks at all of the structures, the RNA structures, and I developed a way to predict what an effective antisense sequence would be to try and block a gene from being expressed. And I then compared it to what other existing programs do, like if one of them is even Fisher Scientific, and based on those percentages, mine hits the percent of what natural target sites hit at like really high percents, and the other ones share a couple base pairs of complementary. And really, it's sort of we're hoping a new way to look at regulating gene expression, and it can be used for a whole bunch of different applications. The private funding was pretty much great, because this past summer gave me the time to focus just on my research. And as I said, I've been trying to do so much work over the academic year, but really there's just no way that I could have put as much effort into this as I could have without this past summer. To actually write the code and sit down and actually go through and check out all these other genes and look through the stacks and find the sequences. And there's just no really way I could have done that without this practice.